scientific method to stop climate change. You'll see that the title is Ultra High Throughput Engineering, but we'll talk a little bit more about that after we talk about these papers. So first, we gotta measure the damage. So carbon dioxide levels have been rising drastically ever since about the year 1950, and for millennia, carbon dioxide levels have never been this high. Of course, these carbon dioxide levels have been rising at a steady pace ever since the Industrial Revolution, as we already mentioned in that project, when people started moving to cities and quit their jobs as rural farmers. Okay, so in our experiment, calcium hydroxide solution was placed into 2 vials. Into one vial, I exhaled for 30 seconds, thus adding carbon dioxide. Then I shook both of them for 30 seconds, and then I absorbed. We found that only the vial to which I exhaled in was cloudy and had a white powder in it. This white powder is calcium carbonate. And therefore, calcium hydroxide is an indicator of carbon dioxide presence in water. Over here, we see the reaction. Calcium hydroxide solution plus CO2. After the reaction, there's no enzyme in this reaction, makes calcium carbonate and water. This is our experiment. Now we're going to turn our attention to photosynthesis. The process of photosynthesis could very well be the key to stopping climate change. The process of photosynthesis is 6 molecules of carbon dioxide plus 6 molecules of water plus light energy makes 1 molecule of glucose and 6 molecules of oxygen. One of the dark reactions in photosynthesis is called the Calvin cycle. The Calvin cycle is a process of carbon fixation which has 3 steps, which will be explained later. Here we have a plant cell and chloroplast. Where are all the enzymatic things taking place? Well, the key to this Calvin cycle and solving climate change is through an enzyme called rubisco. Rubisco is important in the cycle of carbon fixation because this carbon dioxide over here is the one being fixated. And if we can speed up this reaction faster and faster, then all over the world, carbon dioxide levels would go down drastically. Now, rubisco is present in all green plants. But where is it present? It's present in the chloroplasts, the green parts of the plant cell that contain the green pigment chlorophyll. Therefore, the chloroplast is what makes the plant cell green. So, chloroplast hold rubisco. Here's an image of an LOD leaf at 400 times magnification. The diameter is 200 micrometers. Now let's talk a bit about phytoplankton. What is phytoplankton, you may ask? Phytoplankton is better known as algae, which we see all over. Though many people falsely believe that plants are the only things that are fixating carbon in the air, they account for only 30 to 40% of carbon fixation worldwide. Worldwide, 60 to 70% of carbon fixation is because of phytoplankton. Phytoplankton generally takes the form of a greenish-blue color because it also contains chloroplasts. Some different types of phytoplankton are cyanobacteria, flagellates, diatoms, euglanida, coccolithophores, and brown algae. Please do not confuse diatoms with the diatom in the electric field. Diatoms are generally a purple color and circular, which makes them different from other different types of phytoplankton. Now let's talk a little bit about rubisco. Rubisco boasts a size of 10 nanometers, 500 to 1,000 times smaller than a chloroplast. Rubisco is the most abundant enzyme on Earth, and it has a much higher catalytic rate in phytoplankton. Rubisco is also present in all photosynthetic eukaryotes, such as algae, fungi, and all plants, as we've already observed. What are our next steps in this project? Our next steps, as we already explained eDNA on the previous board, we're going to collect eDNA from oceans at different latitudes in collaboration with the National Oceanic Aquatic Administration. Phytoplankton strains vary by latitude and a small amount by longitude. Then we're going to sequence rubisco genes by phytoplankton and then prepare those rubiscos in the lab and test their activities. We're we're going to find the best and improve further by a high throughput cetacean screening. And we're also going to introduce photosynthetic rubisco in nature. So some of our experiments over here are, for example, we have a microscope showing an LOD array.